for giving me the opportunity to speak here. So my name is Engkvist. I head what's called the molecular AI department at AstraZeneca. But we basically develop new methods and try to push more machine learning AI in drug design. So basically, drug design try to answer two questions: what to make next in a drug project and how to make it. It sounds, of course, quite easy, but of course, the devil is always in the detail here. So how do you run a drug discovery project in the industry, or at least in big pharma? It is basically you have something called design, make, test, analyze cycle. So you start, as you can see on the left, with a starting point. It might be a high throughput screening hit, might be a hit from DNA encoded library or virtual screening that have some okayish properties, or at least it's a starting point, but you need to optimize it before you have a clinical candidate. So then you optimize it through what's called design, make, test, analyze cycle. And that usually tends to hundreds of cycles and 3 to 6 weeks per cycle, and takes approximately 3 years before you have a candidate drug. And of course, what we would like to do is to make it quicker with machine learning and AI. And it's basically what comes down to is two things. To the right, in each design cycle, try to make more compounds and try to make the most informative compounds. And what you see to the left, select the most efficient route to synthesis with machine learning AI, as well as much as possible to use chemistry automation. So what are key priorities in machine learning AI is what I will go into more detail: deep learning based de novo molecular designs. We have synthesis prediction, which you don't have time to discuss. The same with more accurate property prediction. And then finally, we would like to make the step from machine learning into AI to do what's called decision making under uncertainty. And to the right, you will see the chemistry automation lab that we set up over at our Gothenburg site in Sweden. We not only try to select the right compounds, but also select the right compounds that we can actually synthesize on the automation platform. So why we are so excited about machine learning in AI for drug design? I think it comes back to a lot of deep learning. I mean, neural network has been around for a long time. Very popular when I started in the industry around 2000. Then it kind of disappeared and was replaced by support vector machine and the random forest. But it's come back since 2012, 2013, first in image processing, and then also in the drug design and drug discovery. And I think the advantage with the deep learning is so flexible. You can put in very many different architecture, as you see on this slide. In the middle, you have what's called a multi-layer feed-forward network. It's common for like activity prediction. You have convolutional network for biological image processing. You have autoencoded neural network. And what you get into more detail, we've used a lot is recurrent neural network. But the information, as you can see to the bottom right, not only flows from the input layer over the hidden layer to the output layer, but you have also information flowing in sideways. So that means that it can very easily or very suitable to learn strings, sentences, or time-dependent phenomena. Of course, that it can learn strings is for something that we see can be used for drug design. So why are we doing it? And why do we really use deep learning to explore the chemical space? It's basically that when we generate new ideas for molecules, we want to sample from the whole chemical space. If you do enumeration, maybe on a good day, you can enumerate maybe 10 billion compounds. It sounds a lot, but it's actually just an extremely tiny fraction of the possible chemical space, which has been estimated to 10 power of the 40, 10 power of the 60, 10 power of the 8. It doesn't really matter how big it is, but it's just that it's not enumerable. So you need to use more probabilistic methods to sample the chemical space. So what we've been inspired to and what we published a few years back together, the collaboration with University of Münster, Marvin Segler, who was at that time working there, is to say there's an analog between natural language generation and molecular structure generation. So if you think about natural language generation, if you train your recurrent neural network on a text corpora, then basically when you start to generate sentences, like the grass is, there's a conditional probability then what the next word will be. It's much more probable the next word is green than the next word is purple, because the grass is green is a reasonable common sentence. So the key is here the conditional probability. When you're in a specific place in a sentence, you have a conditional probability what the next word would be. And then of course it's not so big leap to actually use it for molecules. So on the bottom you see if you have a carbon atom, next to it and you have a double bond, then basically it's a high probability that the next will be an oxygen. So you have an acetaldehyde, which is a very common molecule. And in cheminformatics, we have a very efficient language to describe molecules. It's called SMILES. And in the middle, you see a molecule, and below that is the SMILE string. And basically, so it's a very compact way to describe the molecule. You have the color coding that you can see which actually atom in the molecule goes where in the string. So there, basically, we have the connection into natural language generation and RNN. We can describe our molecules with strings. So that means we can train an RNN on a set of molecules. Could be an in-house database, could also be so to say the Campbell database or some other database. And then we do that, and now I will show so to say basically how we generate it. So we can sample through the first character in the SMILE string. So you see the probabilities to the left. It's the capital O oxygen or capital C carbon have the highest probability, and the probability is written to the right is the log P. So green means high probability, yellowish is white, low probability. And then first you sample the oxygen, and then you will basically the molecule will be generated to the right. So you will see an oxygen to the right on the slide. And then if the first is an oxygen, then you have a conditional probability what the next would be, which next character will be sampled. And you can see the distribution. And if the first is an oxygen, it's a very high probability that the next will be an equal sign, which correspond to a double bond. And then we can see it's basically selects a double bond. And then you can see basically to the left, that is goes step for step through the characters, and it starts to generate the molecule, and the molecule is built up to the right. And this is basically a trick of what we are applying to sample the chemical space. It's basically train an RNN and then sample the characters based on the conditional probability. And as you see to the right, it builds up a perfectly druggable molecule. So that has given us a tool to sample the chemical space very efficiently. But that's not really enough for drug discovery projects, because you have certain criteria in your project that you would like to reach. Just specific potency on your primary target, specific selectivity profile. Hopefully it should be soluble, it should be permeable, and have good what's called absorption, distribution, metabolic excretion properties. So what we are doing is we're running like a design, make, test, analyze cycle in the computer and apply something called reinforcement learning, basically to learn in the RNN which molecules looks good, which look bad. If it look good, those weights get slightly increased. The weights for the molecules that looks bad get slightly decreased. So you can basically through reinforcement learning learn how a molecule should look like. And have a short movie that hopefully can illustrate that. So we like to sort of say design molecules that are similar to Celecoxib, which is a drug on the market. It's in the upper right corner, and it should be molecules that are similar and have a similarity coefficient of 0.7. And you can see the average score evolving on the bottom. So now it starts to generate molecules. This is of course much slower than it does in reality, but it samples around in the chemical space, and then yeah, looks more or less randomly around, then suddenly it starts to find molecules that look similar to Celecoxib, and then through reinforcement learning, it starts to generate more and more molecules that are similar up to a similarity score of 0.7, because that we set as the input. We can of course also set that it should be exactly identical to Celecoxib. So that's basically how we can use deep learning, first to generate molecules, and then through the reinforcement learning technique to generate molecules that are relevant. And then we have a platform that we apply it to. And then besides the design, which I just described, is in the middle, you have to the bottom left, we're also including synthesis prediction, where we have collected data, both internal data and license from the literature, to create a synthesis prediction engine. And to the right is the chemistry automation platform that when we always can, we try to synthesize the molecules on the platform. I will not go into details, but we have run several proof of principle studies internally on live drug design projects, and they have been successful. So lessons learned from project is that we could identify scaffold, completely novel scaffold in a crowded chemical space, and we could actually very efficiently optimize them. We could reach what's called lead optimization phase. We synthesize in less than 100 compounds, which is very unusual. ADME, absorption, distribution, metabolism, and excretion, especially binding affinity, are still limiting factors. That's actually, it's very creative. You need to basically prioritize ideas hard, because you can generate very many good ideas. And it also means that for the chemistry resources in your project, you might front load, so you have more chemists earlier in the project than you usually have. So currently we're running it on 12 to 15 projects over all 3 small molecular research sites at AstraZeneca. Most of the proof of principle studies have been done together with respiratory disease area, but we also now employ it to projects in oncology, in cardiovascular, and of course, we continue to build the platform. I mentioned that binding affinity is a bottleneck. So we are part of a project called Melody, which is an IMI project, where 10 pharma companies will use privacy-preserving machine learning to improve our binding affinity models. So that's something I'm honored to be part of, and leading one at work package. We will really get an answer. If you have 10 times more data, how much can we then improve our models? Finally, just a final slide before I open up for questions. So what a challenge for AI-driven drug design is what we've shown is that it can work, and it's same with chemistry automation, can always work. But of course, there's still a challenge to scale up it to your whole project portfolio. And what is still the challenge is a lot around, and hopefully there are also physics-based models can help, is that how do you apply it really to projects for a lot what's called low data volume, but you don't have that much data. If you have a lot of data, I think we have shown internally, then you can really optimize your compounds very efficiently. Binding affinity and solubility prediction are major bottlenecks. So they also hopefully something that physics-based modeling can help with. Binding affinity, clearly, yeah, from QSCR model, it's the difficult to get accurate that is actually needed to decide which compound to synthesize or not. Solubility is a very important parameter, because if you have low solubility, then everything else is difficult to measure. So it's actually both what's called kinetic solubility from an amorphous phase, but also thermodynamic solubility from a crystal phase. So, and that's been really a Cambrian revolution, new AI methods. So really to actually understand and to assess all of the work that has been is really difficult. So hopefully there could be more, like say, competitions where people can use the methods prospectively. So besides scientific, there's also educational, cultural, and logistical challenge to actually roll out AI and machine learning to a whole portfolio or project in a pharmaceutical company. And I think we have not really shown that we can transform drug design. We have established that it's a useful tool that can be, under the right circumstance, very productive. But there's still some more steps to go, as I just outlined, before you really have transformed the drug design process. Just slides of acknowledgement. To the left is the team. A lot of people there. I've been fortunate to have not only my staff scientists, but a lot of postdocs, PhD students, graduate students, and so on. Funding from the European Union. I was part of a very successful IT and BChem. I had 6 PhD students, 50% of the time, where they made a lot of the development that led to the results and the methods I showed today. And that, I would like to thank you and leave a few minutes for questions.
Ola, thank you. Can you hear me still?
Yes, I can hear you.
Good. So it seems like we can open to anyone who might have questions at this point. Can we see anyone wishing to pose a question? It looks like silence. Is anyone wanting to say something? Who is it?
Andrew.
Andrew Potterton from UCL. Go ahead, Andrew.
I was wondering what method you use to score your generated molecules. Yes, because you have that graph of the different scores. I was just wondering, what did you use? Publicly available scoring method like Guacamole, or did you use sort of an in-house method?
You mean to benchmark the generative models? Yes. So we have been using both Guacamole and Moses in the public, but we have also published our own, which we think is the most, for what we would like to do, the most important, that we sample the whole chemical space. And we have published that several articles where we published our methods and compare it with the GDB database, which is a database that is basically generate all possible molecules up to a certain size through graph theory. So we put a lot of emphasis on the completeness, that we actually can sample from the whole chemical space we're interested in. But for instance, LSTM RNN is performing very well in the Guacamole tests.
You alluded to, this is Ben Limecooler in Edinburgh, you alluded to the problem of community and social and philosophical challenges. I presume you mean in terms of getting acceptance for these types of approaches from the existing players in your industry. Are there scientific developments that would help you to bridge those barriers?
I think that science is still an active field. We are definitely not finished evolving the novel design of property prediction synthesis bricks. So it's an active field, so definitely much more can be done. But I think there's also an educational aspect. We need to think differently in a project from a normal project, that you have much more ideas that you need to assess in the beginning. To generate molecules, you need to also to really think through what you would like to have. So it's a bit of a different thinking as well. And that's definitely education needed. There's also a logistical challenge, because if you want to run, so to say, synthesis quick on an automation platform, you need to see that you have all the building blocks internally, because if you want to order them, you have kind of lost the potential gain by producing automation. So that there are other besides scientific challenges.